I'm Julie Costanzo. I'm working in Strasbourg University in France, and I'm here in the Radiation Research Society to interview Professor Claire Rodriguez-Lafrasse. So Claire, let's introduce yourself a little.
So I'm Claire Rodriguez-Lafrasse. I am professor of biochemistry and molecular biology at the School of Medicine in Lyon. It's the second city and second university in France. And I am also the head of the Department of Biochemistry and Molecular Biology in the Hospital of Lyon. And concerning radiobiology, I created the radiobiology team in 2003.
Okay. I'm really glad to have you here today because I have to confess I worked with Claire a few years ago. So what are your area of research right now?
Yes, we are part of a laboratory of excellence which brings together 7 teams in the region of Lyon. And we are involved in innovative radiotherapy. So my team is involved in carbon ions radiotherapy, hadron therapy, and also in radiosensitizing nanoparticles.
Okay, so your research is more about a cellular level or small animals models? What are you studying?
Yes, we are using both. We are using head and neck cancer models, cellular and animal models. We are also working on glioblastoma and sarcoma.
Mm-hmm. Okay, so where you irradiate with carbon ions?
Yeah, at the beginning in 2003 we irradiated at GSI in Darmstadt, Germany, and next we irradiated in Ganil in Caen. And since 2016, we had the opportunity to irradiate 2 times a year at Chiba in Japan.
Great. Yes. So, and when you irradiate with carbon ions, is mainly cancer cells, or you also irradiate small animals with carbon
No,
ions?
it's impossible to irradiate small animals with carbon ion beam facilities. We only irradiate cancer cells.
Okay. And do you irradiate with protons?
Yes, we have also a facility in Lyon with 3 MV proton beam, yes.
Okay. And what are the interactions with the physicists in your lab?
We provide biological data to physicists. Yes, I forgot to say that my team is affiliated to CNRS, and we are part of the Institute of Nuclear Physics of Lyon. So we have strong interaction with physicists, and we provide data for their modeling. They have a patent on the NanoX models, and this is a model between MKM and LEM, and they take into account the dose deposition, but also reactive oxygen species generation.
Okay, that's great. And you mentioned at the beginning nanoparticles, so you're working on radiosensitivity?
Yes, during this meeting we spoke about the AGUX nanoparticles. This is the first in man clinical studies with EV injection of nanoparticles. These nanoparticles were developed in Lyon by Olivier Timon, who is a chemist, and we did in my lab the proof of concept of their radiosensitizing effect in head and neck cancer model, and also in brain metastases, melanoma brain metastases before the clinical trials. And this is a phase 1 study, and these nanoparticles are well tolerated by the patients, and there is a clinical benefit for 10 out of 12 patients, and we plan to do the head and neck study, phase 2 study, in the next year.
Explain me a little bit more about carbon ions and your area of research.
Yes, we highlighted 5 specific mechanisms involved in response to carbon ions compared with conventional radiotherapy. So the response is independent of the size of telomeres, because you know that the telomeres are highly sensitive to oxidative stress, and patients with glioblastoma are responders to radiotherapy if they have short telomeres, and on the contrary, those with long telomeres are radioresistant. And there is no effect of carbon ions depending on the size of telomere. The second thing is that they are more effective on cancer stem cell. There is also a specific signature, and the loss of a chromosome is a specific signature of carbon ions, and these alterations are not transmitted to the progeny of irradiated cells. And also we showed that there is less metastases, less invasion of migration in response to carbon ions, which lead to less recurrences locally or metastases. And finally, we found that IF1 is a major actor in the response to carbon ions, and it's not stabilized in response to carbon ion and do not trigger metastases, apoptosis, and proliferation.
Okay, so carbon ions are not all about RB, but more than...
Yes, yes. It depends on the energy deposition, which is very different between photon and carbon ions, and the dose distribution carbon ions in related to reactive oxygen species generation, which is quite different between carbon ions and photons.
So you also mentioned your clinical work. So what is it exactly about?
Yes, yes. I'm specifically involved in cancer biology, and we have clinical studies where we are interested in specific biomarkers for the response to radiotherapy. For example, we have a clinical trial in head and neck cancer patients, where we compare the biomarkers expressed in the biopsy and in circulating tumor cells before, during, or after radiotherapy.
Okay,
it's one example. We have others too.
Okay, wonderful. Would you like to add something more?
No, thank you.
Thank you very much for your time.
It was a pleasure for me.
Yes, thank you very much.